And I think it's
Sounds
tough
very
for the
familiar to
students.
where we're at.
Yeah, I feel
Yeah.
bad for the students, especially the grad students. I mean, they were locked out for 2, 3 months and they weren't able to do any of their thesis work. So at least they're in a position now where they can continue to do experiments, albeit on a limited basis for some. But it's something's better than nothing, I guess.
Right, for sure.
All right, I think we still have some people joining, but we might as well go ahead and get started to keep things timely. So I'll turn it over to you, Rob.
Okay, great. So the Department of Biology here at Trent is very pleased to welcome Dr. Costin Antonescu. I apologize if I mispronounced that, but he's an associate professor and director of the biology undergraduate program at Ryerson in the Department of Chemistry and Biology. He completed his bachelor's and PhD at the University of Toronto, and then he did his postdoc work at the Scripps Research Institute in California. So his lab studies the mechanisms that regulate membrane trafficking within the cell. And his lab is interested in studying membrane trafficking to gain a better understanding of diseases such as cancer and diabetes. His lab is currently interested in determining how signaling receptors are spatially organized within the cell, how they're activated, and how they regulate membrane trafficking processes such as clathrin-dependent or clathrin-mediated endocytosis. So again, we're very happy that he's taken the time out of his busy schedule to talk to us today, and we're really excited to hear about his lab's recent work. So I'll hand it over to Costin.
Thank you so much for that kind introduction, and thank you so much for inviting me to be part in this seminar series. It's a real pleasure to be able to do this. It's my first time giving a remote seminar, so looking forward to this as well, and hopefully there'll be some time to ask or to answer questions if there's any towards the end. So before we get started, I think that even though we're all working remotely, at least partly, I think it's important to acknowledge the land that on which this work was done. So I want to start with a land acknowledgement. So Ryerson is in the heart of Toronto, and Toronto is in the Dish with One Spoon territory, which is a treaty between the Anishinaabe, Mississaugas, and Haudenosaunee that bound them to share the territory and protect the land. Subsequent Indigenous nations and people, Europeans, and all newcomers have been invited into this treaty in the spirit of peace, friendship, and respect. So I want to start today with a question that hopefully everyone can sort of dream about a little bit. And I want to ask you, how differently would you behave if you won the daily grand lottery or some other lottery where you get an unimaginable amount of money to do whatever you want? How different would your life be? And if you were sort of on the flip side of things and thinking about trying to understand how people work on or live on a day-to-day basis, how much would you really learn about what people do if you were to study lottery winners? Probably you wouldn't get a very good idea of what every person's day-to-day life is and how they live day to day. And so what I want to suggest today, and this is the reason I'm bringing this up, is that I think there's some parallels here to be drawn between how we study cells and study cell biology and cells and culture, and cells winning the lottery essentially from a metabolism point of view. So this is the point I want to make, that there's a lot of things that we don't quite understand very well yet because we haven't considered nutrients and metabolism and how that actually changes the behavior of cells in some very interesting and specific ways. So what do I mean by metabolism? So just in a very general sense, I'm not going to bring up memories of people from studying this in biochemistry classes of having to memorize all the different steps of glycolysis and the Krebs cycle and all those fun things. So don't worry, I'm not going to bring that up. It's important and super interesting, but it won't be part of this talk. So in general terms, cells have to essentially acquire a variety of different nutrient sources from which they use a variety of different metabolic processes to build up biomass and macromolecules, as well as energy-carrying intermediate molecules like adenosine triphosphate. And so the cell has to constantly have, and every cell in your body does this, has to constantly have a supply of nutrients that allows it to live and carry out its daily or everyday functions. And cells have to have an ability to respond and adapt when this supply is limiting. So this requires cellular adaptation. And this is where I'm getting to with the example of the lottery. when we study cells in culture, typically we don't think about this because we want to keep our cells happy, we want to keep them alive, we want to keep them growing fast so we can do more experiments. But in doing so, we feed them essentially the equivalent of a lottery winning. All the nutrients they'd ever want, and nothing ever is deficient. So I think one of the things that's important to consider is that that's not what cells experience in a normal physiological setting. So if we look in the body, these measurements are sometimes very difficult to do on the scale of individual cells, but imagine in a person or another organism, we have blood vessels, we have endothelial cells here that line the blood vessels, and the interstitial space of tissues, and then epithelial cells here. And so there's been many studies that have looked at measuring how much nutrients, what's the availability of certain nutrients to cells in different tissues. And in the plasma, for instance, in an individual or person that is not diabetic, in the state before a meal, for instance, we're looking at a blood glucose of about 5.5 millimolar on average. But the concentration of glucose in different tissues can actually be quite a bit lower. So interstitial fluid, so within organs, can be somewhere around 3 millimolar, and in the brain can be as low as 1 millimolar. So if you think about what does that mean, does that matter? Well, when we study cells in culture, and this is going back 60 or 70 years of looking at media formulations, usually glucose is in the 10 to 20 millimolar range. So probably close to an order of magnitude higher. And you can make similar comparisons to what happens to other nutrients, amino acids, nucleotides, fatty acids, what have you. So basically the idea is when you're, much of what we've learned about cell biology has been learned from studying cells that are incredibly satisfied. And not to say that that hasn't been really informative, it certainly has. We've learned a lot about this, but it's an important dimension that I think we should think about every now and then in different contexts. So why does this matter? Well, if a cell ever has less than what it needs, the cell has a series of sensors to help it sort of weather the storm. So sometimes a cell will have less nutrients than it wants, less glucose than it wants, less ATP than it needs. So what does it do about it? Well, it can't do business as usual, because if it does business as usual and doesn't have enough nutrients, it might die. And that's not good. So there's proteins, of which one of them that my lab studies quite a bit is AMP activated protein kinase, or AMPK. This is a protein that's made up of a few different subunits here in the middle. It's activated when there's nutrient stress. So whenever there's low glucose or low level of ATP, these signals essentially trigger activation of an otherwise dormant or inactive AMP kinase. The AMP kinase gets activated and rings the alarm that there's not enough nutrients for the cell to do what it needs to do. What does AMP kinase do? Well, it will have many substrates. So it has, on the order of 50 or so substrate proteins. And I won't go through each and every one, but in general, what AMP kinase does is it enhances energy or metabolite yielding processes, so nutrient uptake, expanding the mitochondria, burning fat, so fatty acid oxidation, and it inhibits processes that tend to consume energy, so biosynthetic pathways. It's not about to start building more material if it doesn't have, building blocks to do that. Okay, so that's what AMP kinase does in general. And the questions have been, well, AMP kinase is activated under some pretty severe nutrient deprivation. And it's always been considered sort of the alarm that rings very, very infrequently to signal that something's really, really wrong. But I think what's emerging more recently is that under normal physiological nutrient conditions, AMP kinase is on the verge of activation, right? So cells, when they're normally in the body, are kind of oscillating between activating AMP kinase or not. It doesn't take a dramatic loss of metabolites to do this. So it's not necessarily just tied to catastrophic situations or disease. And the example for this is here. So this is an experiment from the lab that looked at what happens to the activity of AMP kinase. We're looking at the ability of this enzyme to cause the phosphorylation of its substrate ACC, acetyl-CoA carboxylase. I won't go into detail about what it does. It's just a measurement of how much AMP kinase activity there is and how metabolically stressed a cell is when this is activated. So under reducing or lower glucose conditions, we see that around 2.5 millimolar glucose, this becomes activated. as we drop the glucose concentration, which again is the physiological range of glucose that cells see in a tissue. And when we silence AMP kinase, this is lost. So it tells us that this is really what we're looking at here. The cells are responding to lower nutrients and triggering an adaptive response. And this isn't just something that we have found in our lab. There's other papers that have found that this is the range in which AMP kinase is activated, right around between 0 and 5 millimolar glucose, that this is where cells begin to miss what they really want, which is nutrients. They have to start making decisions about how to budget their nutrients, and they need to adapt. So an interesting phenomenon about this is that AMP kinase itself is both sensing the problem and fixing the solution. And so if we look at the behavior of cells under sort of physiological conditions, and we look at individual cells, they actually have a very interesting behavior. So I'm not going to go through the details of this paper, but I'll just explain that this is a way to measure, using a fluorescence readout in individual cells, the activity of this enzyme called AMP kinase. So on the y-axis, you see the activity of the enzyme, and these traces that you see are individual cells that are measured for the activity, right? So in this case, you see relatively flat lines. It means that these cells are perfectly satisfied. There's no activation of this enzyme. If we look at a slight glycolysis impairment that mimics what cells are facing in a physiological condition, what we see is that AMP kinase in individual cells goes up and down, and up and down, and up and down, doing these oscillations. And this is a really interesting thing to keep in mind, because it shows that a normal cell under physiological glucose conditions has a lapse of nutrients, activates AMP kinase, which allows it to adapt. Then it has normal glucose, so it's no longer stressed. But then it finds itself stressed again, and so it goes through these waves and cycles of activation of AMP kinase. So this is actually probably very reminiscent of what cells are doing on a regular basis. They're constantly on this edge of not having enough, having just enough, not having enough, just having enough, and having to adapt on a time scale of hourly adaptation. So similar idea here with just a different way of mimicking normal glucose conditions, so I won't go into that detail. And so this takes us to where our interests in this lie. So I've told you about AMP kinase. I've told you about how cells have to sense their nutrients and adapt as a result. How do they do that? What does that adaptation look like? How do the cells change their physiology in a way that's broad enough so they can survive just not having enough nutrients? You know, for an hour. If you keep them without nutrients for days, they're going to die. But this is a way to sort of buffer that for a few hours. And so we turn our focus to the cell surface, the plasma membrane. And this is something that my lab has had a long-standing interest in, but also one that is really important from a metabolism point of view. So if a cell is going to get more nutrients to make up a deficiency of a certain nutrient, it's going to get it from outside the cell, in part. So a lot of the transport proteins that can allow the cell to recover from nutrient stress are on the cell surface. You know, these proteins here are colorful. They're not really meant to represent individual proteins, but you can imagine that there'd be some proteins here that are responsible for uptake of nutrients. There's also a lot of proteins that spend metabolic molecules like ATP on the cell surface. So it just seems like it's both the source of additional nutrients and a way to shut off the consumption of nutrients when a cell has a lapse in metabolic requirements. So with this in mind, we started thinking about how a cell could do this, and my lab's been interested in membrane traffic for quite a while. So membrane traffic is the formation or the transport of proteins and lipids through vesicle carriers. With relationship to the plasma membrane, this involves the process of endocytosis, which is the removal of proteins and lipids from the cell surface into vesicles. And this is in dynamic equilibrium, let's say, with the process of exocytosis of other vesicles and other proteins to the cell surface. And the important thing to keep in mind here is that these two processes of endocytosis and exocytosis are essentially a way to gatekeep or to control the protein activity of the cell surface. So a cell can change the proteins on its cell surface, by changing the properties of both endocytosis or exocytosis. So I'm going to focus on the endocytosis part in today's talk and how we've been looking at how metabolism and these metabolic cues that a cell has to adapt to change the behavior of the cell by changing this phenomenon here on the left of endocytosis. And before we started working on this in my lab, we had some other studies that told us that this was a good idea. So one of them is the fact that AMP kinase can control the endocytosis and membrane traffic at the cell surface of a few key molecules. So glucose transporters, for instance, exhibit a slower endocytosis when cells are stressed. What happens? Well, if glucose transporters get removed from the cell surface less frequently or at a lower rate, you get more of them on the cell surface. So that's great because if there's more glucose transporter proteins on the cell surface, you transport more glucose into the cell. So under conditions of metabolic stress, that helps the cell survive. And there's other sort of aspects of cell physiology with these other proteins. But we wanted to take a more systematic approach. And I'll tell you a little bit about how we did that and how we started working on a series of proteins, including integrin and others when we get to it. So this was the rationale for this work, that cells can experience different scales and severity of metabolic stress on a regular basis, and they have to constantly be able to adapt their physiology to these conditions. And so we wanted to see how AMP kinase could do that. So the big question is, how do these metabolic signals and AMP kinase cause plasma membrane adaptation? How do the cells change their plasma membrane function in order to account for these really physiologically very meaningful changes in nutrient availability? So I'm going to tell you about three parts. I'll spend a little bit more time talking about points 1 and 2, but really it's about how AMP kinase regulates the cell surface proteome, so the full collection of proteins at the cell surface, and clathrin endocytosis, how this may impact the regulation of integrins, and how all of this matters because it affects processes like cell migration, which are obviously great interest in both in normal physiology as well as in pathophysiological conditions like cancer. And I'm going to talk a little bit about another way to link metabolism and membrane traffic, which is through this O-glucnac molecule that's a post-translational modification of certain proteins. So our first approach to this, when we started working on this back in 2012, was to look to see if we can actually use a systematic approach. And instead of looking to see, does AMP kinase change the function of this particular protein or that particular protein, we wanted to take a screening approach and look to see if we can find what are the proteins in an unbiased way that AMP kinase can regulate at the cell surface. And so we used a strategy where we can use this chemical probe that is cell impermeable, so it can't pass through the plasma membrane. And it reacts only with proteins that are exposed to the cell exterior, so cell surface proteins. And this allowed us to attach a biotin group on here. And using some purification techniques, this allowed us to use mass spectrometry to identify the proteins at the cell surface. And we use this strategy then to see what changes when the cells are exposed to different nutrient conditions. So the cells that we use are an epithelial cell origin, these ARPE19 cells. What's advantageous about them is that they're spontaneously arising. They're not transformed or immortalized with a virus gene. So they actually have a better representation of a normal metabolic state. And they're able to quite readily activate AMP kinase. Here we're using this drug, which is an agonist of AMP kinase. So we're not actually inducing metabolic stress, but we're treating the cells with a drug that mimics the activation of AMP kinase. So we're really able to just look at what do the cells do in response to this alarm going off, right? We're not dealing with all the other possible stress that the cell could be experiencing in terms of metabolism. We're just saying, if you ring the alarm, which is this AMP kinase enzyme, what does the cell do? And what we found using this mass spec approach was that, I won't go through the details of all this, I'll just draw your attention here at the bottom, that when we activate AMP kinase, we identified over 600 proteins that were selectively depleted from the cell surface, right? So AMP kinase is causing a big change within 90 minutes of activation of the proteins that are found on the surface of a cell. So it's completely remodeling the cell's plasma membrane protein complement. And there's lots of things in here. I won't go through each one and all the different examples of proteins we've considered. But what really struck us early on was this network of proteins that are involved in cell adhesion and migration. And our interest here on this one protein, this is the gene symbol, but it's integrin beta 1. So this is a cell adhesion molecule that's involved in linking the extracellular matrix and the actin cytoskeleton. I'll come back to this protein a little bit later in part 2 of the talk, but really we became interested in integrins and cell migration for this reason. And just to show that this mass spec technique wasn't was valid and meaningful, we looked at doing the same sorts of measurements by other approaches. And in this case, we're focusing on integrins. We can label using fluorescence microscopy the integrins that are selectively at the surface of a cell. So what you're looking at here, the brighter that an individual cell is, the more integrins it has at the cell surface. So in a control cell, we see a certain amount of integrins on the surface of these cells. And when we activate AMP kinase, this is decreased on a per cell level. And we can deplete or silence AMP kinase, and this phenomenon disappears. So it really tells us that this is something that the cells are doing in response to this alarm of AMP kinase going off, and telling the cell that there's a metabolic need that needs to be, to which the cell needs to adapt. So we became interested in this notion of how is it that AMP kinase can control not just integrins, apparently, but many other proteins on the cell surface. And what we know from the point of view of regulation of membrane traffic is that endocytosis is able to regulate many different proteins, because many different proteins undergo this phenomenon where they're packaged into vesicles from the cell surface by endocytosis. And in the process I'll talk about in a little bit more detail now, Clathrin endocytosis recognizes proteins that have these particular motifs. These are protein motifs that are found in membrane proteins that are packaged into vesicles and regulated by Clathrin endocytosis. You don't really need to focus on the numbers here. I just want to show you there's several thousand proteins that are subject to this regulation. So bottom line is this seemed like a good way that you could have the cell regulate the abundance of proteins at the cell surface for many proteins at the same time. So it kind of fit with what we were thinking about in terms of what AMP kinase was doing. So you know, if you've taken cell biology, or maybe you are, some of you may be quite more familiar with it than the cell biology course, but if you've taken cell biology, you may have seen the process of Clathrin-mediated endocytosis. I won't go into a whole lot of detail about this, although I'd be happy to talk on at length about it. But I only have 45 minutes, so I'll stop there. The phenomenon is based on this protein Clathrin here, which is shown in blue. Clathrin's special ability is it can form these lattice-like structures. And this ability of Clathrin to form this protein scaffold is linked to the ability of the cell to form these membrane buds that eventually lead to the formation of vesicles. So we call these Clathrin-coated pits because they're pits in the membrane that will eventually give rise to vesicles. And what these Clathrin-coated pits do is they concentrate the certain proteins from the cell surface into the pits that will allow these proteins to be selectively removed. So over the years, there's a lot that's been learned about Clathrin endocytosis. I've certainly been working on this for quite a while myself, as have many other research labs around the world. And we know that Clathrin doesn't do this alone. In fact, there's probably about 100 or so proteins that are working alongside Clathrin to make this all happen. Here's a few of them here that are listed, and they're basically in this process where the budding of the membrane progresses and then eventually leads to the pinching or scission of this vesicle from the cell surface. We can talk about a few sequential stages of this phenomenon, and people have, or scientists have mapped essentially the role or functional requirement of a whole bunch of these different proteins at the bottom at different stages. The good news is I'm not going to ask you to remember any of this. I just wanted to give you a sense that it's complex and, more importantly, that it's subject to regulation, right? So things can essentially progress to a certain stage and then stop. right? It doesn't necessarily need to go all the way to the end. So the key thing here is that clathrin isn't necessarily, as you may have seen it in a textbook, where once it starts, it gives you a vesicle. This phenomenon is essentially can be regulated and can be stopped or aborted at different stages, depending on what the cell's trying to do. And so just another way to say that is this is what you may have seen as a processive process that just kind of goes and you don't think about it. But in reality, this is maybe 40% of clathrin structures that form. And in a cell, you may have more than half of them, depending on how you measure this exactly, where the process begins but then turns over because the cell has made a decision to stop. Something's regulating this and says, you know what, we started, we don't need this, stop what you're doing. And then the bottom line of what I'm saying here is that there's many different points of this assembly where you can have a turnover or either making the process more efficient or less efficient to control what the cell's doing at its cell surface. So then the question is, are any of these gates controlled by AMP kinase, right? So this seems like a lot of different points at which a cell could sense something and do something about the endocytosis of cell surface proteins in order to then change the protein complement of the cell surface. And so to study this, one of the major techniques that my lab has used is microscopy. So this is a form of microscopy called total internal reflection fluorescence microscopy. I'm going to spare you some of the details about how this works, but what this allows us to do is to resolve only the fluorophores that are near the surface of a cell based on the optics of the microscope. And what that allows us to do is if we put a green fluorescent protein on clathrin, we can actually follow the dynamics or the life cycle of individual clathrin-coated pits. So what I showed you on the last slide here in cells. So when you see these little individual dots, it kind of looks like the nighttime sky. But each one of these little dots is an individual clathrin-coated pit that's forming, doing its thing, and then disappearing from the cell surface. And so what we were able to do with this in collaboration back when I was a postdoc with the lab of Gaudens Danuser, now at UT Southwestern Medical Center, using computational tools to not just look at individual structures and try to get something from them, but really systematically use software that can detect individual clathrin structures, track them over time, and use essentially the computer to tell us what's going on with these different structures. Okay, so the whole reason we do this, again, is to try and get a sense of, is clathrin assembly and the formation of vesicles being regulated under different conditions, right? We can now look at this systematically across the whole cell. So the first thing we looked at was some pretty simple measurements of, is clathrin assembly being regulated just by looking at the fluorescence intensity and therefore size of individual clathrin structures. And so when we activate AMP kinase with this drug here, I'll just remind you that this is the drug that causes activation of this metabolic stressor. This is what clathrin-coated pits normally look like on the surface of a cell. Again, you can see these little individual dots are all individual clathrin structures or clathrin-coated pits. I hope you can see that this image is a lot dimmer, right? And so what this means is that, and following the same methods of quantifying, what we've been able to see is that the individual clathrin-coated pits quantified here are dimmer and smaller than they would be normally. So when a cell activates this nutrient stressor, it changes how clathrin assembles at the cell surface and makes the vesicles and pits smaller. And what we've done even further is we've used some of the more sophisticated analyses of the software to be able to resolve how this impacts a few different stages. So the very early stages, the assembly of these structures, eventually the scission. And what I'll just do is I'll show you some of the types of data we get from this, but I'll summarize essentially what this means here is that without going through all these individual data panels, what we've noticed is that the rate of initiation of these clathrin-coated pits goes down in a nutrient-stressed cell. Clathrin structures themselves are smaller, but they are less likely to become abortive, so they're more likely to actually form a proper vesicle. So what does all this mean? Well, it means essentially that this is the two different conditions that we're looking at. Just to summarize, in case I went through some of those previous slides a little quickly, when we have a cell that we consider to be normal, that we're growing under normal cell culture media conditions, this is the equivalent of winning the lottery for the cell. Again, lots of metabolites. It forms clathrin structures that are big, and they're not as efficient at forming vesicles. When we activate AMP kinase under metabolic stress conditions, the pits and the structures are smaller, but they're more likely to lead to vesicle formation. So if we're thinking about what this means, you kind of have the Hummer, which you can use because you've got lots of nutrients or gasoline. Not sure why you would want to, but let's say you do. And then you also have the smart car that is able to operate when there's not a lot of nutrients, so not a lot of gas around. It's much more efficient. So that's the way we're thinking about it here. This is the way the cell changes and adapts to these nutrient conditions. We became interested in what is the mechanism that can allow cells to do this. So what is linking AMP kinase, this alarm, into changing the cell surface protein complement? How does it change the formation of clathrin-coated pits? We went back to our mass spectrometry screen that allowed us to look at the types of proteins that are changed at the cell surface when AMP kinase is activated for clues. And we found a series of proteins called, well, they're collectively proteins that regulate a family of proteins called ARF GTPases. So these are a family of GTPases that are well known to function in regulating vesicle traffic and transport. So we just to give a little bit of background to this, GTPases are proteins that have an on and an off switch. So in this case, when the molecule is bound or ARF is bound to guanosine triphosphate or GTP, it's considered to be in the on state. It can also be in the guanosine diphosphate bound state, and this is in the off state. And ARF gaps are proteins that essentially turn off these ARF GTPases. I won't go into detail here, but I'll just show that it's been known for some time that ARF GTPases control the formation of vesicles and are involved in membrane traffic at a variety of other intracellular locations, including on endosomes and in the endoplasmic reticulum. So this seemed like a good place to start for looking at the cell surface. We were able to collaborate with the lab of Anne-Claude Gingras, with Jeff Hesketh, a postdoc in her lab, to apply BioID to ARF6. This is work that they did in their lab. Without going into a lot of detail about this, BioID is a way to attach a protein domain called BIR-A to a protein of interest, in this case ARF6 that we're looking at. And it allows you to then biotinylate or modify with a biotin residue proteins that are within a narrow radius of this domain, so about 10 nanometers. And that allows you to identify, essentially, what proteins are near this protein. So if we're looking at a possible way to regulate clathrin, we would expect ARF GTPases to reveal an interaction or proximity to clathrin by this method. So in this case, you carry out the biotinylation reaction and then do mass spectrometry to identify what proteins were modified with this biotin. And in fact, that's what we saw. I didn't introduce a lot of these proteins, but I'll just mention this one here, which is clathrin itself. So CLTC is the gene name for clathrin. And there's a few different mutants of ARF6 here that were examined. Basically, the darker the blue and the bigger the circle, the more interaction there is between ARF6, this ARF GTPase, and clathrin. So we can see that ARF6 is big and blue with clathrin, so they do interact. And the ones below it are all of clathrin's friends that I alluded to that interact with clathrin. So really, it kind of told us that ARF6 was in clathrin structures at the cell surface. We were able to use a fluorescence biosensor to reveal that AMP kinase changes the GTP or activation state of ARF6. Briefly, the way this works is it relies on FRET measurements. This involves the resonance transfer of energy between a donor fluorophore and an acceptor fluorophore. So basically, if these two molecules that these fluorophores are attached to are far apart, they don't have any FRET. You don't observe any FRET. You just observe fluorescence. Whereas if you have these two molecules in close proximity, we have fret, and it actually changes the lifetime of the fluorophores in question here. And so all of this is essentially allowing us to measure whether ARF6 is GTP bound or not inside cells. And what we're looking at here, so essentially by measuring lifetime, we measure how much GTP bound ARF6 there is. And the higher the value, the more GTP binding there is. And so what we're able to see here is that, sorry, the other way around. So the higher the value, my apologies for that, the higher the value here, the less GTP binding there is. And what we can see is that comparing the red and the blue, that the presence of the AMP kinase activation increases the lifetime of this fluorophore, which tells us that ARF6 is more GTP bound less GTP bound than the basal state. So AMP kinase regulates and decreases the amount of GTP binding of ARF6. We were also able to look at the way in which AMP kinase could be triggering regulation of this protein from clathrin structures. And so what we're able to look at here is now in two different colors. So we have a red fluorescently tagged clathrin, similar to what I showed previously, but now it's red. We have a green fluorescently tagged ARF6, and we can look to see if the two are found together. So if ARF6 is detected within the clathrin structures that make vesicles, they are. But when we activate AMP kinase, ARF6 essentially disappears, and we can quantify that here. So this is the amount of ARF6 found within clathrin structures. All of this tells us that we have a mechanism that might involve AMP kinase activation, eventually doing this really important job of regulating the cell surface by decreasing the amount of ARF6 bound to GTP. And this led us to ask what's missing here in the middle. And I'm going to, if you'll allow me, because I realize I'm running a little bit slower than I anticipate, I'm going to skip to the end of this section and summarize our summary for section 1, just so I can talk a little bit about section 2 as well. What we were able to find is this protein ARF-GAP3 is our candidate that might be regulating or bridging this signaling pathway. So just to summarize section 1, I didn't show, sorry that I skipped over that piece of data, but what we're finding, our model currently is that AMP kinase increases the activity of this ARF-GAP3, which depletes ARF6 from clathrin, which causes this regulation and adaptation of the cell surface proteome in response to nutrient stress. The bottom line of all this is that this is something that was previously thought to happen just under complete catastrophe, when the cells don't have nutrients under some weird once-in-a-lifetime situation. But really what we're putting forward is that this might be something that's constantly being triggered as cells have to deal with limiting nutrient availability. So by now you might be asking yourself the question of, okay, well, who cares, right? This seems like a nice little trick that cells can do. Change the assembly of this protein I've only barely heard of before. Why does this matter? And that's why I wanted to get to this part 2, where there really is this mechanism does really clearly impact what cell behavior can do. So that has to do with how AMP kinase regulates this family of protein called integrins. So integrins are proteins that are transmembrane proteins. They're made up of an alpha and a beta subunit. They are able to bind extracellular matrix and the actin cytoskeleton on the inside of cells. So this is the outside of cells and the inside of cells. And they have a very important role to play, for instance, in cell migration. So you can see here that integrins link the extracellular matrix in the inside of the cell, and they kind of act like the wheels on a tank, or the I don't know what to call it on a tank, but they're able to sort of allow the cell to have traction as the cell moves in this direction across an extracellular matrix. And the ability of the cell to migrate is really dependent on these integrins undergoing vesicle traffic. And what we were able to look at is that when we activate cells with AMP kinase, again to mimic this metabolic stress, the distribution of integrins on the cell surface changes dramatically, from being sort of more even and spread out to really being focused into individual puncta and dots. And through some analysis, we were able to actually identify that this causes an increase in the amount of integrins in the vesicles, the clathrin-coated pits and the vesicles that derive from them from the cell surface. So really what we're looking at is, I told you before that under nutrient stress, you make the clathrin-coated pits smaller into these smart cars. But really what it is that you're able to cram a lot of cargo, like integrins, into these very smart and efficient vesicle carriers. We also looked at how the pathway that I talked about in the previous slide, which leads to the activation of ARF-GAP3 and the lowering of ARF6 GTP, impacts cell surface integrins. So the overall effect of AMP kinase activation on integrins is to reduce their availability on the cell surface. And we were able to show that this pathway is essentially very similar to how we saw regulation of clathrin that affects the regulation of integrins. So if we look here at control cells, this is cell surface integrins that I showed you previously. We see less of them on the cell surface with AMP kinase activation. And this is prevented when we remove this ARF-GAP3 protein from cells. And last bit of data that I want to show is what this is impact in terms of cell migration. So I mentioned integrins are involved in, or the regulation of integrins involved in allowing cells to migrate as the cell crawls over a surface. What we're able to see here is cells that are crawling, in a normal situation. And we see that cells that have been activated with AMP kinase to mimic this metabolic stress are pretty arrested. I think you can even pick this up just by looking at them. And we can also quantify this here by showing the mean velocity of cells. All these different treatments are treatments that can trigger metabolic stress. So really what we're looking at is the ability of this pathway to change the cell surface protein complement of any given cell. But in this case, it really does matter because it impacts the ability of the cells to migrate. So it's a really important phenomenon as the cells tune their response to changing nutrient availabilities. So I'll skip this part here and basically just summarize by the fact that our working model is that this regulation of AMP kinase through these GTPases, we think impacts this particular protein called DAB2. This allows it to then regulate integrins, but there's potentially a similar regulation of a variety of different cell surface proteins that are related to integrins and that can explain back what I started talking about, how AMP kinase changes not just one protein, but multiple protein activities at the same time at the cell surface. We think it might have to do with the fact that this is a new signaling pathway that links metabolism to a metabolic adaptation of the cell surface that's constantly occurring inside cells. Unfortunately, just in the interest of time, I won't have time to talk about part 3, but it's essentially the bottom line of it is it's a different parallel mechanism that cells can sense different types of nutrients and similarly change their cell surface protein activities. So I'm happy to talk about it. Anybody wants to talk about it afterwards if there's any questions, but I'll skip that and get to the take-home message, which is something that I think is going to become more and more important in biology, is to understand that lots of what we study is context specific, and the differences between different studies, if you don't always agree in terms of the findings, that can actually be an important source of information. So the plasma membrane is a highly adaptable organelle that undergoes dynamic remodeling based on metabolism and nutrient availability. We have been finding that clathrin endocytosis is a metabolically sensitive pathway that's gated in multiple different ways by inputs from metabolism, that the metabolic control of endocytosis impacts signaling, migration, and maybe a lot of other things inside cells. And maybe this is a time to think about carefully about how cell culture conditions that lots of us use have maybe can be enhanced by studying the nutrient composition to see how that impacts our whatever favorite processes that we're studying. So with that, I want to make sure to thank the people whose work I've highlighted here. So Christian de los Santos, Laura Orafiama, Nicole Leschishin, Sadia Rahmani, and Gizem Genk, who are graduate students and postdocs in my lab that contributed more directly to this work that I had a chance to share today. And most of what I've talked about today is funded by NSERC, and we're very fortunate with these other funding sources as well. Many thanks to all of them. So thank you very much for listening, and hopefully there's still a few minutes for questions if there are some.
Well, thank you very much, Dr. Antonescu. I personally enjoyed your talk very much. So normally what we do is we kind of open up a chat now. So anyone feel free to unmute themselves and talk. Otherwise, we can, if you want to write in the chat, we can help facilitate asking questions. But while people work up the courage to ask, I have a few questions for you.
Great.
So first of all, are there multiple genes that encode AMP kinases in mammalian cells, or is there just one?
Great questions. There's multiple genes. So I mentioned that it was a trimer that made up of an alpha, a beta, and a gamma subunit. There's 2 alpha, 2 beta, and 3 gamma subunit genes. And they're similar, but not identical in their function. So part of where cells now begin to have different responses to metabolic availability in different tissues has to do with different cells in different tissues have different isoforms that are expressed at different times. So that's one of those sources of isoform specific responses that we still have to really understand much better.
Another follow-up question. So in that sense, with all of the different proteins that are regulated by AMPK or AMP kinase, is a knockout lethal then?
Great question. So the catalytic subunit of AMP kinase is the alpha subunit. You can do a knockout of the alpha 1, you can do a knockout of the alpha 2, but if you do the 2 together, it's embryonic lethal.
Okay.
So it would definitely be consistent with this sort of very fundamental role of sensing metabolism very early on in development and probably throughout adulthood as well in most animals.
Okay.
I have a couple of questions.
Go ahead.
I guess they're sort of related. The first is with the proteome work, the RP19 cells. Did you grow them, or do you grow them in like a nutrient-rich media, like DMEM, or you sort of alluded to this at the end. Are you able to drop out certain nutrients and see what effect it might have?
So when we start considering individual nutrients as individual parameters to experimental conditions, it starts to get really complicated really quickly. Much of the work that we've done with RPE cells involves just regular media that is higher in glucose than physiological. More recently, we have been working with the same media that's identical except for not having glucose. And there we can control just adding back either glucose or glutamine. That's where we started. Some other labs have done some pretty important work to try and devise more physiological media that's available as well. So that's more nutrient limited in glucose and other amino acids. But I think we've got a long way to go before we can actually ascribe certain concentrations of certain nutrients to individual effects. I think it's going to be a lot of experimental biology and a lot of mathematical modeling that's going to help with that.
Yeah, you'll have to tease out the different variables and the different components. And I guess that sort of leads into my second question, which is, have you looked or considered downstream what might be happening? So how the autophagy pathway might be fitting into the regulation by AMPK?
It's the running joke that I'm sure my lab is sick of hearing me say, but I like to think of us in some ways as superficial. So we look at the cell surface and we don't go too deep into the cell, although that's definitely not the case. We have projects that look at things that are happening at the lysosome and the nucleus and things like that. So we haven't looked at autophagy. We know that some of this regulation is not due to induction of autophagy because we can do manipulation like knocking down ULK1, and we still see some of this regulation. So I think those are important things that we need to do and keep in mind. But given the fact that we have such a massive triggering of internalization, one of the destinations that could be happening downstream is to feed this into some sort of autophagy pathway to further enhance nutrient availability. So that'd be something to look at for sure. That'd be a good idea for sure.
Awesome.
I have another question. So can you explain, I'm sure you mentioned this, but I probably missed it, with your biotinylation experiments where you're able to basically identify proteins at the cell surface. Can you explain how, what's your readout for that? How are you identifying those specific proteins?
Great question. I went over that super quickly, so apologies for not explaining that more thoroughly. So the way that we do this, it's a chemical reaction at the beginning where we provide the cell culture with a probe that is impermeable to the cell, so it can't go through the plasma membrane. It can only react with primary amines that are exposed to the cell exterior, so transmembrane proteins that have an extracellular domain. We can then kill the cells, lyse them, and do a streptavidin pull down. So basically that allows us to pull down the chemical group that we use to label the protein. So it pulls down only the proteins that were at the cell surface. We release them, and then we identify the proteins by mass spectrometry.
I see. So it's whatever is bound to those individual receptors.
That's right.
Okay. Gotcha.
I have a question. This is Carolyn Capron. Sorry, my video is being a bit glitchy, my internet, so I just turned my video off. But so I have a question about the first picture that you showed about that showed the distribution of the beta-1 integrin, and it looked very much like it was in cell-to-cell contact regions. And it reminded me of some really old research that said that some integrins are involved in cell-cell adhesion interactions.
Yes.
It was even, I think even the first one that you showed right at the beginning, even more so. Is that like, do you think that is happening, or is that just the pictures that you happen to select?
So we see it pretty clearly that integrins do line up at sites of cell-cell contact. I think to do this carefully, we would have to measure volumetric considerations. So just in the way that the cell shape occupies space, I think we'd have to exclude that this is a volumetric artifact of looking at an enhanced labeling at the edges of cells. But I think there's probably going to be some enrichment of integrins at sites of cell-cell contact. Whether integrins are playing a role in that, we haven't really looked at it in a tremendous amount of detail in these cells. So we'd have to look at it. If we look, for instance, at cells that are super confluent, so they would have lots of cell-cell contacts, the regulation of AMP kinase of integrins tends to change a little bit. It's not as pronounced. So what we may be looking at is a completely different function of integrins in a confluent monolayer of these cells than in a condition where these cells are able to migrate more freely. So that's a great question. That's a long-winded answer for me to say that I don't know, but it's an important question and one we should probably look at.
Yeah, it does. It certainly. I mean, the standard role is cell matrix adhesion for the integrins, but it doesn't. I mean, doesn't mean that could be the only role, and it could play into the differences in cell migration that you saw as well, because it seemed in a way that those cells were not the mutant cells were not migrating, but also the little movement that they did, they weren't showing that contact inhibition that migrating cells often do. So they were, if they touched, they just stayed, instead of in the controls they move away from each other.
Mm-hmm. Yeah,
anyway, it's very interesting. Yeah.
Well, thank you.
Any other questions for Dr. Antonescu? No? Okay. Well, thank you so much for giving us your time this afternoon. We really appreciate it. I definitely found your talk very interesting, and yeah, we appreciate you taking the time